First, for those of you who are not familiar with ENCODE, it is an international consortium founded by the National Human Genome Research Institute. There are many collaborators. Here is a list of the main collaborators. And also the aim of this project is to build a comprehensive atlas for the functional elements in the human genome. And the functional elements that ENCODE looks at are includes the hypersensitive sites, meaning the open chromatin sites, and also the DNA methylation, his modifications, and transcription factor binding sites, and also the RNA polymerase II occupancies. And recently they added the long-range interactions between chromosomes, as well as the enhancers, repressors, silencers, and insulators. Here is a cartoon showing the data ENCODE has collected. It is basically organized into cell lines and the assays that have done with the cell lines. ENCODE has the initial phase of ENCODE characterized 3 cell lines, and then they expanded gradually over the years, and now there are almost 400 different types of cell lines. And for each cell line, they have collected different types of functional elements, and there are also a whole bunch of ChIP-seq experiments for each cell type. This is a timeline for the ENCODE project. Starting from 2003 until 2007 is the pilot phase. There are 3 cell types, and they annotated 1% of the genome. The technology they use are mostly array-based. Starting from 2007, it's the production phase. They mostly adopted the next generation type 247. And at the end of this phase, they claim that 80% of the functional portion of human genome has been annotated, and the data they collected are looking at over 2,000 protein coding genes and over 18,000 RNA genes. Here are some numbers. So the data produced from the pilot and production phase are hosted as a UCSC genome browser. Starting from 2012, they expand the production phase. They now there are more than 300 cell types, and also there are almost 300 antibodies. And they also started looking at some new functional elements such as the RNA binding proteins, silencers, enhancers, and the long-range chromosome interactions. And the new batch of data are now hosted at the ENCODEproject.org website. So here is a screenshot of the UCSC genome browser. Here is the ENCODE data at the UCSC genome browser. You can basically browse the data from a data matrix. It's organized at cell type level and also the assays, and there is extended data matrices for the ChIP-seq data set. To download the data from this UCSC browser, you can go to the download link here, and then they categorize the data into different genome build. And also here you can see different assays. The problem with this website is that first you can only access data through their FTP links, and also the metadata is not very clear. Yeah, so for example, you can get the metadata from their cell type or antibody list, but it's not very specific for each individual data set. And when I process this batch of data, I basically parse the file name of the file names, and then get the information mostly from the file names, such as the assays and the replicates, etc. And as for the new website, ENCODEproject.org, it has a lot more functionality for us. So this is the new ENCODE data website. It gives more filtering functions for users to select from. For example, we're interested in a ChIP-seq data set, and we can choose different organisms, organs, and treatment, and also the data types. So, and also when you click on a specific data set, if you're a regular user, it gives you a well-organized summarization of the metadata. And if you want to see more information, you can look at in the JSON format, where there are a lot more information associated with each experiment. And also, the data hosted at ENCODEproject.org can be automatically accessed through the REST API. The metadata are organized in JSON format. So this is a data model example of their data model, and you can find the full schema on the GitHub site. So here is an example of what I've done with the ChIP-seq data from ENCODE. So there are a few steps. First, I download the ChIP-seq peak files and also the metadata. So ChIP-seq, the raw data for ChIP-seq are just the FASTQ files, and then you map these reads to the reference genome and then identify the significant peaks. So I download the peak files and also match each peak file to their metadata. Now I know the cell types and the. So after I match the peak files to metadata, these peak files are in the BET format, meaning that for each peak there is a chromosome coordinates. And then I take these coordinates and map them to genes, meaning that I look for the genes that are close to this peak. And then I generate gene list for each of the experiments. I did the most of the steps using in-house scripts in Python and Java. And here are some numbers for the data set. So for the new batch of ChIP-seq data, we have both human and mouse samples. And we have over 600 hit modifications, as well as some CTCF and RNA fold2 occupancy. So this is how it looks like after I generate a gene list for each experiment. So this is just an example. This is actually the ChIP-seq data from the pilot and production phase of ENCODE project. So basically, this is the first column is a label for the gene list, and it is the transcription factor in the. So this is the transcription factor, this is the cell type, and this is a list of genes that are associated with the peaks from the ChIP-seq experiments. Here is a summary of the data sets we have. So these data can be accessed through Enricher. It's a web app developed in the lab. So far we have, the number of experiments are the data set we got from ENCODE, and then after I merged the replicates, unified experiments, there are this many terms for each of the data sets. This is for the transcription factor binding sites. This is for his modifications. We also have some additional his modification data from the epigenomics roadmap project. And we also have some ChIP-seq data from CHIA, which is a manually curated database. And then we will add these new batch of data soon. So here is example of how to use these data from ENCODE. So here is a figure from a study we did last year. We collect all these seq data, and then we create a network consist of transcription factors and histone modifications. We also connect them through the histone modifying enzymes. So we would like to take these transcription regulome and start to look at the gene expression data from the L1000 experiment and try to identify the upstream regulators for the up and down genes. And that's it.